Cellular injury occurs when cells face stresses that exceed their capacity to adapt, disrupting their normal function and homeostasis. Cell injury progresses from a normal state through reversible changes like membrane blebbing and organelle swelling to irreversible damage with membrane destruction and nuclear changes. Cellular injury can be caused by various factors. Physical agents include trauma, extreme temperatures, and radiation. Chemical agents like toxins and drugs, and biological agents such as viruses and bacteria can also damage cells. Nutritional imbalances, hypoxia or reduced blood flow, and immune reactions are other important causes of cellular injury. Hypoxia or oxygen deficiency affects cells in multiple ways. It decreases cell proliferation, alters organelles like mitochondria, disrupts endocytosis, and can increase cell migration. One key mechanism of cellular injury is ATP depletion. This reduces energy-dependent processes, affects sodium-potassium pump function, and leads to cell swelling and calcium influx. Membrane damage is another critical mechanism. It involves phospholipid destruction, loss of membrane integrity, increased permeability, and release of cellular contents. As seen in this microscope image, progressive damage leads to visible changes in cellular structure. Other important biochemical mechanisms include DNA damage with mutations and strand breaks, disruption of protein synthesis through ribosomal detachment and dysfunction, and oxidative stress causing free radical damage to cellular components. Cellular injury exists on a spectrum from reversible to irreversible changes. Reversible injury features cellular swelling, organelle alterations, and membrane blebbing. If stress continues, irreversible injury occurs with lysosomal rupture, membrane destruction, and ultimately cell death. Cells can adapt to chronic injury through various mechanisms. Hypertrophy involves an increase in cell size, while hyperplasia refers to an increase in cell number. Both are adaptive responses to increased functional demand. Other adaptation mechanisms include atrophy, metaplasia, and dysplasia. Metaplasia is a reversible change from one differentiated cell type to another, often in response to chronic irritation. An example is when bronchial epithelium changes to squamous epithelium due to cigarette smoke. When cellular adaptations fail, cells undergo death via one of two main pathways: necrosis or apoptosis. Necrosis is uncontrolled cell death characterized by membrane rupture and inflammation, often caused by ischemia or toxins. Apoptosis, on the other hand, is programmed cell death that eliminates damaged cells without causing inflammation. Recent research has made significant advances in our understanding of cellular injury and death. Ongoing studies focus on inflammaging, which is chronic inflammation associated with aging, CRISPR gene editing technologies, cell therapy advancements, and single-cell analysis. Ferroptosis, a recently discovered iron-dependent form of cell death, has gained significant research attention. Studies suggest that caffeine may protect against neuronal injury by inhibiting ferroptosis and reducing oxidative stress.